Today we're going to look at the nervous system, which is really something that could use a lot more time. I'm going to just try to touch on what I think are the important things and prepare you mainly for the assignments you have as much as I can. I was able to yesterday get caught up on the grading, and of course people were doing work yesterday, so there's a few more things that I have to grade, but I'll be able to get on those pretty quickly. I wanted to give people a good idea of what they have in the course right now, grade-wise. Now, it's a little odd. I don't know if that's the right word, but what I've done in the past is printed out a grade report. I have a function in the grade book, part of Blackboard, which will print out all of the assignments for the course and what your score is. And really all it is what you would see by clicking on the My Grade section in Blackboard. So I haven't done that because it wouldn't give you any information you can already just get that way. And in fact, the report that I would print out would be missing one important aspect. If you go into My Grades, next to the makeup assignments, if you were here that day and on time, you're exempted from that. And the only way you can tell that is in the My Grade section, you'll see a gray circle, which stands for exemption. If you hold the mouse over that gray circle, a little word will prop up saying exempt. If you don't see that gray circle, that means you're not exempt, or I haven't gotten around to marking down the exemptions. But I think I am up to date through last week. Obviously, not this week because we're just starting, but for everything but the nervous system makeup assignment, it should correctly tell you whether you're exempt from something. If you feel that you should be exempt and you're not, just feel free to email me. I'll check my attendance reports. It could be that you feel that you shouldn't be exempt because you were here that day, but you're not exempt because you weren't here on time that day. The makeup assignments count for people being late or absent. So I'll just verify that with my attendance records. It has happened in the past that people have said, I know I was here and on time that day. Why am I not exempt? And I just double check it and it's there. One time that happened because on my grade book thing where I can check off the exemptions, it doesn't display everybody on one screen. I have to scroll down to see the last couple of people. And so I just failed to hit the exemptions for those last couple of people. Just because I didn't scroll. So mistakes can be made. Just feel free to double check on that. And I'll get back to you with a specific response. Now, one thing I have done with printing out these reports, which I don't think was entirely useful, is I can go in and, well, under My Grades, you see at the end, actually I don't know what it looks like to you in the My Grade section, but on the report it does it in order. At the end of the report, which I assume is at the end of the My Grades page, it will say lecture score, lab score, and overall grade. The way that the grade book works right now is it only counts things that have been completed and graded. So a lot of assignments are auto-corrected, so as soon as you complete it, the score is there. If you're doing things late, those auto-corrected ones The scores held up until I can get in there and check them off, which I've done on any of the autocorrected things, part 1, 2, 3 assignments, up to this point. I did them just before class. So you should see all of that stuff if it's been completed. And then any of the part 4 or other assignments that I have to grade by hand, those are not counted for your grade until I've gone through them. Anything that hasn't been completed or that hasn't been graded, at least as far as the grade book is concerned, isn't counted in what you see. The score that you see is just on the things that you've completed. What I've done in the past is I can switch the grade book so that we'll take anything that hasn't been completed and count that as a 0. And when I've done that in the past, the point was to kind of, I don't know, scare people into understanding that the grade you see right now is not necessarily the grade you're going to get. You have to complete the rest of the work. Somebody at this point in the semester, maybe suffering from fatigue, might see an A or a B there and say, oh, I've got this made, I don't really have to worry about it. But then they don't complete the rest of the work for the semester and they end up with an F. Okay. I often get texts or emails at the end of the semester saying, I don't understand why I got the grade I did. It said I had a B or something like that. When I start to actually grade things and I switch it so that it counts things that are incomplete as 0s, that can really bring grades down. So I've, in the past, printed out 2 versions of the report, one that looks like what you'd see right now if you went into my grades, and one that would show what it looks like if you haven't completed things. For people that haven't completed work, they're behind schedule, they're a couple of chapters behind, or maybe they haven't done anything, whatever. That can be a bit of a wake up because they can see, oh, I haven't done anything. I have a 3 in this class out of 100, or something like that. And hopefully that can get people motivated. Now, it's a little late in the semester to be at that point. You need to have everything completed by next Thursday. Not this Thursday, but next Thursday. So if you haven't completed work and you're putting it off till last minute because you're like, oh, I can do it next week, no problem. You won't be able to do it after Thursday next week. Also, keep in mind when you're doing things late, I have to check them off because you've submitted them late. I don't get on things immediately, okay? So if you do part 1 of a chapter and you're waiting for me to check that off to go on to part 2, you might have to wait a little bit until I can get into the grade book and check things off. And then once I do that for part 2, you might have to wait. Once I do that and you complete part 2, you might have to wait a little bit until I can check that off so that you can go on to part 3. Okay. It's highly unlikely that you can get through an entire chapter's worth of stuff if you're doing things late within a day, just considering the time delay in me checking off assignments. So if you're behind on any chapters, please get to them as soon as possible. If you're behind on multiple chapters, do all of the part 1s and wait for all of them to be checked off, or do the part 1 of the first one, and while you're waiting on that, work on the part 1 of the second one, et cetera, et cetera. Just try to maximize your time. I just haven't found a very effective way to get this message across to everybody. There are people that will put things off to the last minute, and you'll get caught. Honestly, people that aren't keeping on top of the work throughout the semester aren't going to do very well. When you're putting things off to the last minute, the struggle to get things in on time usually hampers your ability to perform at your best. Now, that's more directed at people that are just really procrastinating. If things have come up and you can't get to things on time, but you're trying to keep on top of it for, the past couple of weeks, you've been doing work that you let slide and you're on top of things, then you're probably not the type of person I'm considering. But people that are thinking, okay, next Tuesday I'm going to come in to do the practical, and then Wednesday I'm going to sit down and do all the work I haven't done online. That's your mode of thinking. You're not going to have a very good time of it. And you probably are not going to respond to me saying this either, if that's your mode of thinking. I don't know necessarily if anybody's really in that position, but I want to try to get that idea across. Before I go on to talking about things, I want to describe an assignment that's a little bit different from what you've been used to. So we're currently in the Unit 9 folder, the last material for the semester. The next folder is the Lab Practical 2 and Cumulative Final. The first 3 items here are the outline, cases, and online assignment for the second practical. It's exactly like the first practical, except of course it's over different material. This is muscle and nervous system stuff. The musculature and nervous systems historically have not really been entirely separate systems. Really, the skeletal muscle system is controlled by the nervous system. So these really do kind of come together a bit more than other cases that we've had. The first 2 cases are really directed at the musculature. The third and fourth cases are directed at the nervous system. That fourth case is going to kick your ass, okay? That is the hardest case you'll ever have from me, trust me. Spend some time thinking about it, reading the questions carefully. If you don't understand a question, please ask me for assistance. Trust me, okay? There's just certain questions in that assignment that people habitually get partial credit on because they're not reading the entire question. So be thoughtful, look for help when you need to. I got a text from somebody yesterday, I can't remember if it's this class or AMP2 class, but somebody texted me yesterday saying, I've been trying to complete part 3 of whichever chapter, and I just can't do it. And my response was, why didn't you ask for help? Okay. That's my job, is to help you. Okay? If you're working on an assignment and it's giving you trouble, don't just throw your hands up in the air and say, I can't figure it out, and give up. Ask for help. I will give you some help. I'll give you annoying help. You'll ask me a question, I'll ask you a question back. But the question I ask back is designed for you to think about or get closer to the answer. And if it doesn't help you, then you can ask for more help, and I'll help you a little bit further. Okay? But please do take advantage of that resource in the course. The other thing in this folder is the cumulative final. What we're doing next week is just the practical. It's not the final exam. We do the final online. It is a 50 multiple choice question test. Like other multiple choice question tests on the Blackboard site, it is time based on 2 minutes per question. So if it's 50 questions, it's a 100-minute timer, or an hour and 40 minutes. Honestly, that's more than enough time to complete this kind of test. If we were doing it in class, which I used to do, we would do the practical and the cumulative together in an hour and 50-minute period, okay? Which I'd round up to 2 hours. Which would mean you would take a 50 multiple choice question test, which is the practical, and a 50 multiple choice question test, which was the final, in 2 hours, allowing really an hour for each. Oftentimes, people would spend an hour and a half on the practical and only have a half hour left over for the final. Because that time period doesn't really work well to allow for people to put their the proper effort into both of those tests, I split up and did the final online. In doing that, I was able to set up the final in kind of an interesting way. It's made up of the multiple choice questions that you've been getting all semester. Since it's on Blackboard, I can just tell it to use the same pools of questions for part 1 and 2 assignments that you've had all semester. Now, when I say it's the multiple choice questions you've had all semester, I just mean the part 1 and 2 assignments. I don't mean lab quizzes and that sort of thing, although I ought to put those in there. I won't for you, but maybe sometime in the future. Each assignment, part 1 and 2 assignment, has a pool of 20 to 30 questions. And when you've been taking those assignments in the past and you open it up, you get 10 questions. Then you take it again, you get 10 different questions, that sort of thing. What am I trying to say? I just rebuild the assignment each time. In the cumulative final, it's kind of set up the same way. There's 50 questions. I don't quite remember. I think it's 5 per chapter or something like that. Might be 4. So for that 4 or 5 questions, I tell it to use the pools for part 1 and 2 of that chapter, okay? So, which is between 40 and 60 questions total between the 2 pools. So of the 4 or 5 questions for that chapter, you're going to get randomly pulled out of those pools the questions that you see. The likelihood of 2 people having the same set of 50 questions in the final is minuscule, practically nil. There's 300 some odd questions to populate 50 questions in the test, and the likelihood of the exact same 50 questions being pulled in 2 separate attempts on the test, it's just astronomically impossible. Not impossible, because it is a random procedure, but highly, highly unlikely. And even if the same questions were pulled for everybody, the answer choices usually get randomized too, if you haven't noticed that. You answer a question once, what was A, B, C, and D? The second time you take an assignment, you get the same question, the A, B, C, and D choices have been randomized. There are a few that doesn't happen. Sometimes the order is important, like it'll be, a progression of numbers, and instead of randomizing them, it always goes like lowest to highest or something like that. So those don't randomize, but the vast majority of them do. So even if 2 people were to get the exact same questions, the answer choices wouldn't be in the same order either. Okay. Where was I going with that? To prepare for this, it's actually pretty straightforward. I don't want to use the word easy because it does take some effort, but all of the questions you're going to get on that, you can see before you take the test. If you go back into the attempts that you've made on the part 1 and 2 assignments all semester, you can see those questions. Now, you might not see all of the questions in the pool. If you wanted to, you have to retake the assignments and see more questions pulled out of the pool. Now, this semester, I've done things differently. You're getting an average score over all the attempts, and opening up one attempt that you already have, I don't know, an 8.5 or a 9 on, isn't the greatest idea. I don't quite know how this will work because it's the first time I've done it, but what I'd like to do is, if you are to the point where you're going to study for things, text me and say, or email me, whichever works better for you, and say, I'm done with such and such assignment. Any further attempts you make on that assignment after you let me know that you're done with it, I will not count towards the score. I'll have to manually go in and erase them when I do the grading, but I'll have a message from you with a timestamp, and I'll know anything that you did up to that point goes towards the grade. anything after that point won't count. It will affect your grade as you see it because I have to manually go in and erase those things, but I'm not going to do that until after you complete the final, so that you have those questions there for reference and grading. But so if you did introduction part 1 and 2 already and you have those to the score you want, but you only took it like twice and so you haven't seen all the questions, and to prepare for this you want to see more attempts, then just send me a message saying I'm done with those assignments, I'm about to start studying for them. I'll acknowledge that and you can go ahead and start doing it, and I will know anything from the timestamp on that message you sent back are the only grades that count. Okay. It's sort of a trade-off in this averaging thing. It used to be that whatever your final score is the one that stood, and so people, when they're studying, they would just take, open up several attempts, and as long as the last one was a 10, that was the one that went in the grade book. But so, like I said, this is the first time I'm doing it, but if you send me a message saying you're switching to studying for those assignments, I will not count any further attempts towards that grade. I'll erase them when I go to grade stuff. So, does that make sense? All right. I actually need to double check, and so while I'm saying this, I'll open up the test options. But it's supposed to allow you to take the test 3 times. And yes, it does. So I've got the multiple attempts thing selected, and it allows for 3 attempts. So you can go in and take that test. If you, say, get a 38 out of 50, and you decide to take it again, and you get, say, 42 out of 50, and you say, well, I can do a little bit better than that, you take it again, you can improve it. The best of the 3 attempts is the one that goes. So if that 3rd attempt after you get a 42 is a 39, that doesn't mean that you get the 39, you get the 42. Okay, that's the one that's going to go into the grade book. Basically, if you didn't take, if you do worse on a subsequent attempt, the best grade stands. But once you get a 50, there's no point in taking it again because you can't do better than a 50. The first time I ever did this online, the first student who took it, she took it before she came in for the final practical, and she had a 50. So I just, as she finished and handed in her test, I grabbed her real quick and I said, I noticed you got a 50 on the cumulative final. How'd you do that? And she said, it's exactly what you said. I went back and looked through all the questions. I knew what all of them were, and I could just sit down and take the test, and no big problem. Okay. And she did that in 1 attempt. So it is entirely possible. It just takes a little bit of effort. And the point of that effort is to have you go back and look through things from the beginning of the semester, which is why it's a cumulative final. But so you have that. Yeah, so the due date is August 6th at 11:59 p.m. The 6th is the Thursday of next week, right? Yeah. Our class is on the 4th. So you have until the end of the day to complete it, okay? Either at midnight that night or as soon as I get up on the 7th, I'm going to go in and shut down your access to Blackboard. You have to have everything done by then, okay? You have no idea when I'm going to do that, because if I'm up late that Thursday night, I might just go ahead and get on and turn off access right then. Or if I go to sleep before midnight, then I'll do that as soon as I get up in the morning when the dogs have to go out. Okay. Any work that you haven't completed, you have up until basically midnight on the 6th to complete. Now, they have other due dates, so if you're doing them late, you do have to deal with the fact that I have to check them off. Well, if you're doing chapter assignments, you have to deal with the fact that I have to check them off. If it's something else and you do it late, if you do it at 11:59 on the 6th and you don't see a score for it, I'll go through and check it off when I do the grading and it'll be fine. But if you're waiting to go on to complete part 2, 3, or 4 of a chapter, you're not going to be able to do that because I have to check it off for you to move on. But I've kind of already covered that, I think. You can go back and redo anything. If you want a better score for one of the part 1, 2, or 3 assignments from any chapter, just go back and do them. If you're going to do that, you're going to bring the assignment average up. Do them in reverse order. Okay? Do the part 3 assignment, because once you do that, since it's late, it won't let you go on to the next assignment. You can't redo part 4s. So do the part 3, and it'll block your access to part 4, which you should have already done. Then go back and do part 2, okay, if you want to improve that. You'll notice when you do part 2 that the part 3 assignment will disappear from the folder. The grade won't disappear. Well, the grade will say it's waiting to be graded, but it won't erase your attempts. It'll just block your access to the thing. Okay, so for instance, this week's folder right now, you see part 1, but you don't see part 2, 3, and 4. You have to complete this to see part 2. You have to complete part 2 to see part 3, et cetera, et cetera. If you have all of the assignments visible because you've already done them, when you do one again because it's late, which of course wouldn't account for this folder until after next Tuesday. If you do an assignment after you've completed and it's late, whatever's dependent on that score will disappear. Doesn't erase the attempt, just removes the link off of the page here. So if you open up part 3 to do another attempt to bring that average up, part 4 will disappear. Okay. Doesn't erase the attempt, just the links disappear. So if you're going to redo any assignments, please do them in reverse order. Then you don't have to wait for me to check off part 2 before you can do part 3 or something like that. If that doesn't make sense to you, I'll sit down and explain it to you individually, but it's an important part of doing that part of the course here at the end. Any questions about wrapping things up here at the end of the semester?
So the question is also if you write the 6th,
Say that again?
The practical will also do by the 6th.
Right. So if you go into that folder, that assignment's open and available, you should start on it as soon as possible. Today I will have gone over the material that you need to prepare for some of those questions, especially the 4th one. It's about some stuff for today. So if you've already looked at the online assignment for the practical and you have no idea what case 4 is asking about, that's because we haven't covered some of it. But after today, you should be prepared for the most part to deal with that stuff. So, other questions?
So there's 2 parts of the practical and the online too.
Right. So the practical is just like last one. There's the in-class portion, which is what we're doing next week. And then there's this assignment here, which is the online questions, just like you did for the first practical. And yeah, I'm pretty sure that this is set. Its due date, yeah, it's August 6th at midnight also. Okay. There is a chance that I won't be shutting Blackboard down at midnight at the end of the 6th, but you don't know, don't count on it. Don't say, oh, I'll go ahead and do that stuff at 2 in the morning. Because if you get there at 2 in the morning, it's not there. I warned you. It might be, but you can't count on it. So, any other questions? Oh, does remind me next week, since all we're doing is a practical, there's no point in us being here at 8. Okay. So what I've traditionally done for that last week is we start at 9 o'clock. Parking will still be on its own schedule. So if you're getting here just in time for 8 o'clock and you're driving around trying to find a parking space, an hour later, it's going to be worse. Okay. So if you want to be sure that you get parking the way you do most every week, then show up here for 8 o'clock, but we won't start for 9. That'll give you an hour to cram the last little bit of whatever in your head. Or if you want to, get a little more sleep beforehand and you come in for 9 o'clock, just understand that you're probably not going to be able to park too close to the building. Okay. I suppose they have the lots across the street open. Oh, they've got the grass open.
Yeah.
Okay, that's not so bad. It used to be that people have to park down the hill, but I guess because they're doing that construction out there. All right, so the grass isn't so bad. But yeah, so next week I'll have the window on the door covered. So you'll wait out in the hallway, and when at 9 o'clock I'll open the door and we'll get started. You have an hour and 50 minutes for that practical, so we'll be done by 11 o'clock for sure. And if you're done earlier, then that's all we're doing. You're welcome to leave at that point. So, other questions before we move on? Wasn't really planning on taking up a whole half hour, but these are important things to talk about at the beginning of the course. Okay. So I am going to try to move through this week's material somewhat fast, just to make sure we hit on things, all the important stuff. Last week we looked at what nervous tissue is and how it creates electrical signals. I got to the end of what I wanted to there. I kind of had to wrap it up. Wait, actually, did we get out early last week? Oh no, that was A&P too. So I got to the end of that probably right at the last minute, but I did cover what I wanted to, including neurotransmitters and synapses and that kind of stuff. So we're going to move on to anatomy of the nervous system, which is the focus of this chapter's assignments. But really, the following chapter, called the somatic nervous system, deals with a little bit of it also. So this chapter, anatomy of the nervous system, is obviously mostly about anatomy, but it's really impossible to completely separate anatomy and physiology. So in talking about the anatomy of the system, I also cover some things about the physiology. And then the somatic nervous system chapter, which follows that, has essentially 3 parts, technically 4, because it has an introduction. But then it has sensory perception, central processing, and motor responses. The sensory processing, sensory perception section is actually a topic for A&P 2. So the central processing and motor response part of this fits in with what we're talking about with the anatomy chapter also. You can skip over the sensory perception section if you want to. Technically, it's considered part of the reading assignment, but we will be concentrating on that in A&P 2 for the most part. But be familiar with the other 2 portions of this chapter, because this is sort of where we deal with that. I will also point to, although we will not spend much time on it at all, if I can squeeze it in the last 5 minutes, I'll mention it. This chapter here that says the neurological exam, usually I have a 3rd week to do nervous system stuff, and so I spend that 3rd week looking at the clinical ramifications, neurology type stuff, which is what that chapter is about. There is not a set of chapter assignments for that chapter. I just include it as a way to understand neurophysiology a little bit better. And I'll talk about that at the end if I can. But there's no assignments based on that. It's just helpful to understand more what's going on here. Now, I usually spend a little bit of time going through what's the embryological perspective, which by a little bit of time, it usually takes about 20 or 30 minutes to get through this stuff, sort of tell the whole story. I don't have time for that, so I just want to hit some highlights. The nervous system is rather complicated at the adult level. So it's nice to start at its most basic level and work your way up. And that can point out some of the relationships between things that help you understand how it functions in the adult system. It starts with just explaining how the embryological structures start to give rise to the nervous system, which is the outer covering in the embryo, what you would rightly assume is going to make up the skin of the human being this is all about. Eventually, actually part of that tissue that's destined to be the skin is also destined to be the nervous system. And it puckers in a little bit and it forms a tube that runs along the posterior side of the embryo, just underneath the outer covering. That's what we call the neural tube. It's a tube. It's a hollow tube. It's kind of like, you know, one of those balloons that you can fold up and make animals with. And that's the nervous system at its simplest. It has an anterior end and it has a posterior end. The anterior end becomes the brain. The posterior end becomes the spinal cord. And so to understand the brain a little bit, you can look at how it develops. And there are two snapshots of neural development that help us understand its structure a little bit. When the anterior end becomes a brain, basically what happens is the tube kind of balloons out a little bit into what are called vesicles. And the first snapshot we look at is when we see the primary vesicles, and the second snapshot is when we see the secondary vesicles. This picture shows both snapshots. This is the primary vesicle stage, and this is a secondary vesicle stage. There's a picture of sort of a longitudinal section laid out here, and then a lateral view of the intact system here. So when we talk about vesicles, imagine this originally was just a tube, and so it's kind of bulged out here, and there's a little bulge here, and there's a bulge here. Those are the three vesicles. At the primary vesicle stage, there's three vesicles. They have Greek names for them. All three names have encephalon in them, which is really just the Greek word for inside the head, meaning the brain. Cephas means head in Greek, so encephalon means inside the head. The prefix for each of the words basically tells you what part it is. Prosencephalon is referring to the forward part. Pro means at the beginning, basically in Greek. So prosencephalon in English really directly translates to forebrain. And then mes means middle. And so the mesencephalon is what we call the midbrain. So forebrain and midbrain are pretty much direct translations of what those Greek words mean. The third one, rhombencephalon, does not actually translate directly to hindbrain. Rhomb means something else. It's actually describing its shape. But in keeping with forebrain and midbrain, hindbrain's the rest of the brain. So those are the three parts. Then these three vesicles continue growing. The prosencephalon, or forebrain, develops into two different vesicles, and the rhombencephalon, or hindbrain, does the same thing. The mesencephalon doesn't get any more complicated. So what we call the midbrain at the primary vesicle stage is always the midbrain. Okay, and really it's a small part of the brain in the end, and that's because the forebrain and the hindbrain kind of grow out of proportion to the midbrain and take up a lot of the space. The forebrain's two vesicles at the secondary vesicle stage are called the telencephalon and the diencephalon. The telencephalon gives rise to what we call the cerebrum. When we think of the brain, when you see a brain, I've got a bunch of models here, and I'm going to have you grab these models a little later on to look at as we're talking about structures. And that usually think of this big gray wrinkly structure on the outside. That's the cerebrum. What we're actually looking at here is the cerebral cortex, the gray matter on the outside surface. That's only 1 millimeter or 2 thick, and right underneath that's a bunch of white matter, and deep inside the white matter is more gray matter, nuclei that are part of this cerebrum. But that's the cerebrum. It's this big part right here. This is what the telencephalon develops into. The diencephalon, if we look at the adult brain, is this portion right here. Okay? So I can cover it up with kind of the first segment of my finger. Okay? It's about that big. The telencephalon is hard for me to cover up with even just my hand. Okay? It's a huge portion of the brain. And one of the things that really distinguishes the human brain from other organisms is the size of our cerebrum. I just read an article yesterday, and it was a very brief article, but it was kind of interesting. Apparently some data that was recently described compared human brains to monkey brains, and there's a part of the two brains which are obviously different, and it's actually kind of obvious looking at the brain by itself, but they got some functional information, and part of what that part of the brain appears to do has a lot to do with how humans are very different from other primates behaviorally. So that's the cerebrum. The diencephalon is made up of mostly structures that have the word thalamus in their name. Primarily there's the thalamus, but then also there's the hypothalamus and the epithalamus. And then there's another structure which I hopefully will get to mentioning later today called the subthalamus. All of those things are in the diencephalon. We don't always use these names for those things. Like the epithalamus is more commonly referred to as the pineal gland, but its formal name is epithalamus because it's part of the diencephalon. Also, the diencephalon gives rise to what in the embryo are eye cups, and we can see that little eye cup right here. That's the nervous tissue that's going to be part of the eye, namely the retina. So the retina actually develops directly out of the central nervous system. Some ways of thinking about the eye actually considers it a central nervous system structure, but that's a very confusing issue that I don't want to get into right now. But it does start its life as part of the central nervous system. An important part of thinking about the embryology here is to understand this sort of thing. The eye cup originally was part of the diencephalon. If you look at what the retina directly connects to in the brain, it connects to the thalamus and the hypothalamus. Now, it doesn't connect to the epithalamus directly, but that's a human thing. Well, not necessarily human, but it's kind of a mammal thing. In lizards, for instance, there is a direct connection to the epithalamus. There's actually a lizard whose epithalamus is exposed to light and acts as a third eye. The pineal gland in humans and primates is responsible for regulating day-night cycles. So it eventually gets information about whether the sun's out or not, whether it's daytime or nighttime, and it helps regulate that aspect of our behavior. So in the lizard, it has a third eye, which is just its epithalamus, which is directly exposed to light. And if there's sunlight, it knows that. For a lizard, that's very important, because lizards, being cold-blooded reptiles, need to warm themselves in the sun. And so they have
basically a hole on top of their head that they can tell when there's sunlight and they can move into it and stay warm. But this is human anatomy and physiology, so forget about that. The eye cup is connected in the human to the thalamus and the hypothalamus. Basically, the thalamus is responsible for conscious vision, and the hypothalamus is responsible for unconscious or subconscious visual processes. And by that, I mean primarily the regulation of day-night cycles goes through the hypothalamus to the epithalamus, and also regulating the size of your pupil. Okay? If I move over here and I stand in the path of the projector, that bright light there is potentially too bright for my pupils, for my retina, so my pupils will constrict, and that's under hypothalamic control. Okay. Any visual information that elicits an emotional reaction also is handled through the hypothalamus, which is a subject for AMP2. The midbrain is still the midbrain. What it does is interesting, complicated, and a little bit beyond what this course is about, but we have that there. The rhombencephalon separates into these two structures here called the metencephalon and myelencephalon. The metencephalon actually differentiates a bit more than that. We don't look at another vesicle stage. Instead, we just know in the adult, there are two structures that developed out of that. One of those is the cerebellum. So this brown thing on the back of the model that I'm showing you here is the cerebellum. It develops out of the metencephalon. It's about 10% of your brain. It's a pretty big chunk of your brain. Cerebellum means little cerebrum in Latin. So this looks like a little version of this. They're both wrinkly gray structures on the outside of your brain. That's where the name comes from. Adjacent to this cerebellum, right here, this is the pons. Okay? And I'm going to have all of you grab one of these models later on, and while you have it in front of you, I'll explain what these different parts are. But that's the pons right there. I said that a second ago, what I have my thumb covering up, that's the diencephalon, the thalamus and hypothalamus. What I have my other thumb covering right now is the pons. So the space in between the two is the midbrain. Okay? It's a really small part of the brain. Okay? And then everything below the pons is what's called the medulla. Up there, its full name, medulla oblongata, but outside of Adam Sandler movies, people don't really call it that. Nobody get that reference? Anyways, there's nothing else in the brain that's referred to as the medulla at this level. There are some structures maybe that say that, but it's nothing that you talk about outside of like a graduate school setting. So we just refer to that as the medulla. Now, the Greek words that I ran through real quickly here, from the primary brain vesicle stage, we can just refer to those by their English equivalents: forebrain, midbrain, and hindbrain. And then at the 5 vesicle stage, we don't really use any of these Greek words except for diencephalon. Instead of telencephalon, we just talk about the cerebrum. Instead of mesencephalon, we still use midbrain. Metencephalon, depending on which part, we talk about the pons or the cerebellum. Myelencephalon, we talk about the medulla. So really, the only Greek word in this picture that you need to remember is the diencephalon. There's not really a better word for that part of the brain, so that we still use that. But everything else, we just use the English or adult equivalent for each of those structures. So I pointed out the idea that since the eye cup, what's going to be the retina, develops out of the diencephalon, it lets you know right away that the retina is going to be connected to the diencephalon directly, which is the case. The other thing to get out of this is the cerebellum develops out of the same part of the brain as the pons, and that's where the majority of the connection of the cerebellum to the rest of the brain is found. It does connect a little bit to the midbrain, a little bit to the medulla, but it's mostly connected to the pons. If anything needs to communicate with the cerebellum, it needs to go through the pons. Okay. And in fact, the cerebrum needs to communicate with the cerebellum, or the spinal cord needs to communicate with the cerebellum. It can only do that by following the map that we have here. From the cerebellum, information has to go through the diencephalon, through the midbrain, to the pons, and then into the cerebellum. From the spinal cord, it has to come up through the medulla, which can get directly to the cerebellum. You can look at the adult brain and think, well, the cerebellum and the cerebrum are right next to each other. Okay, so you look at this, and they seem to be right next to each other. So wouldn't it make most sense for them to just communicate directly? That's not the case. If this part of the cerebrum right here wants to connect to this part of the cerebellum right here, while in the model they're only a millimeter or so apart, the connections have to go through the diencephalon, midbrain, pons, and into the cerebellum, vice versa. In fact, in the skull, there's some connective tissue that comes off of the skull that sort of wraps around all the brain, and one piece of that goes in between and makes a little shelf between the cerebrum and the cerebellum right here. So there's no physical contact between the two. They're separated out by a little piece of connective tissue. And this map here through the embryonic brain tells us where we should expect any connections to travel. And in fact, that's true. Cerebellum goes through the diencephalon, the midbrain, pons to the cerebellum. That's just the way things are laid out. The embryonic picture here can give you a little bit of an understanding of how things actually relate, because once we get to the adult situation, it gets much more complicated. So use this as a reference to come back to looking at those sorts of things. Now, I'm not going to talk about the spinal cord development a whole lot, because it kind of stays in a tube layout for the most part. But it does get a little bit into that the anterior or front part of the spinal cord tends to be associated with motor responses, and the posterior or back part is mostly about sensory processing. And I'll show you something later on that goes to that. I don't quite remember why this title here, Relating Embryonic Development to the Adult Brain, is right here. I must get into that later on, which is kind of about the eye cup, diencephalon, cerebrum, cerebellum stuff I just talked about. But it does start off by pointing out something that's kind of important to think about. Humans are very different from other mammals, and I use mammals because we all have the same basic neurodevelopment. If you look at a dog or a rat or a mouse, any other type of mammal, you'll see all the same structures essentially in the nervous system, cerebellum, diencephalon, midbrain, etc. They'll look very different because they're each adapted to that particular organism's needs. But also in the embryo, for all mammals, the layout is pretty standard. But then in the adult human, it changes because we're bipedal. We're standing upright. So in this picture here, just looking at the dog next to the human, anterior and posterior is one continuous line through the dog's neuraxis. So its brain is anterior, its tail is posterior. The spinal cord probably ends somewhere in here. I don't actually know where exactly in the dog. But for the human, anterior is thought of as being, kind of at the facial end of things. Posterior is at the bottom of the spinal cord. Okay. So looking at this anterior view of the spinal cord, nervous system, the end of the spinal cord right here, this is the posterior most point of the human nervous system. If we travel up and then we turn forward, this is the most anterior part, because it kind of follows that curve there. little weird thing about humans because we're standing upright. If our nervous system was laid out the same way as four-legged animals, we'd have to be looking straight up like that, or we'd have to be down on all fours. So, just to point that aspect out. This table sort of lays out the primary vesicles, so neural tube, primary vesicles, secondary vesicles, adult structures, how they all kind of match up. The last thing here is about the ventricles. That tube that we started with was hollow. That hollow space in the tube is found still in the adult brain. I think it was must have been last week, I pulled out this thing here, right? Showing you gray versus white matter. We can also look at this and see in the middle of the brain, there's this, let me put it this way, this kind of Y-shaped space in the middle. That's the space left over from the hollow neural tube. And then the thickness of the brain around that is just how that wall is grown out a little bit. Those are called the ventricles. And I want to finish that thought, so I'm going to move to a different part of the chapter to show you pictures of the ventricles a little bit. But this table kind of shows where they're all kind of connected, related to the embryonic development. So I'm actually skipping over a section, which I'll come back to in a second, but module 4 of this chapter, it says circulation and the central nervous system. And this is where I can show you the ventricles. So let me go down to that stuff real quick. Okay. I thought I had a picture of this. I don't. So we actually have some models. Actually, we have this exact model right here in the back, which I'll point out later on. This is what's called a ventricular cast. The way this was made, the original that this model was made from, is plastic was injected into this empty space in the brain and allowed to solidify, and then all the brain tissue was actually removed, and what was left over is this shape that we see here. Here's a drawing showing what that cast kind of looks like, and here it is superimposed in the brain. I'm not going to go to this picture completely, but this is sort of looking down on the same thing from on top. So there are these two big pieces that we can see sticking off to either side, which are these big curved parts here that come to a little point and then come around here. There's one on either side, a left one and a right one. Those are called the lateral ventricles, and that's the empty space within the cerebrum. And in fact, it's what we see, Y-shaped structure that I pointed out here. These are the lateral ventricles. So obviously there's midline right down the middle. The two arms of the Y reaching off to either side are part of the lateral ventricles. They curve through the brain and come all the way around. It's really kind of hard to see because it's such a small point, but right here, and I guess right here, is more of the lateral ventricle, which are curving around. You can't really see it. For the sake of that picture, we're kind of cutting through at this point. So we can see the front part, that's the arms of the Y. And then in the back, we just see a little bit, maybe right about here, that we're cutting through, and it just doesn't stand out as well in the preserved tissue there. Then in the center here, this is what's called the third ventricle. Now it sounds like we're starting to count with 3. It's not true. The first and second ventricles are the lateral ventricles. We just don't actually number them. They were numbered at one time, and I guess it just became politically incorrect to say this is number 1 and this is number 2, because they're pretty much equal. So 1 and 2 here, and then 3, which is really spaced right on the midline between the 2 halves of the diencephalon. There's a little hole right in the middle of the ventricular cast because there's a little nervous tissue right in the middle that crosses over. But other than that, right on midline at the diencephalon, there's some space, which makes up the third ventricle. When I look at this stuff, I usually kind of imagine this to be like a head. The plastic casts that we have, like this one, it's a little bit, looks a lot like a rooster to me for some reason. So that little hole in the middle would be the eye, and then it comes to kind of a point here, actually 2 points that kind of look like a beak. Okay, so it's like this is a head. And then the neck, which is kind of obscured by the lateral ventricle here, is just a tube that goes from the third ventricle to the fourth ventricle. So the fourth ventricle is where it kind of widens out right here, and this is the third ventricle. So the tube that we see right here, going all the way down here, is not a ventricle itself. It's what's called the cerebral aqueduct. And it passes from 3 to 4. The cerebral aqueduct goes through the midbrain, and then 4 is basically at the level of the pons and kind of the medulla. The bottom of the picture here, what we see at this point, is going to continue down on into the spinal cord, and it's just a very thin canal, which I pointed out last time. The cross-section of the spinal cord that I was showing you, the neurons and the ependymal cells in. The ependymal cells line the edge of the ventricle system, and in the spinal cord, they line the central canal. But all of this space, if we looked at the cells that are right next to it, it would be lined completely with ependymal cells. Here's another picture kind of showing it superimposed. So here's a lateral ventricle going through the cerebrum, pointing all the way back here and then coming into the side. The third ventricle is right at the level of the diencephalon. The cerebral aqueduct, which we can see leaving the third ventricle here and continue on here, is passing through the midbrain. And then where it kind of opens up, this is the level of the pons. And then the medulla is down here, and it continues down into the spinal cord. Now what this section is saying, while there's not really a picture like that in the book here, this is showing the circulation of blood into the central nervous system and then the cerebrospinal fluid. While there's blood vessels passing all the way through, passing through all of the nervous tissue, it's sort of separated off. Blood doesn't just directly go into the tissue. There are astrocytes associated with the blood vessels, and we have the blood-brain barrier. So instead, there are certain places where blood is filtered to make cerebrospinal fluid, which then becomes the interstitial fluid of the brain and circulates all through the brain. What it does, like the interstitial fluid in any other tissue, is help to carry waste products away. And then those waste products will eventually get into these veins. There's one right along the top center of the brain, one kind of deep in the center of the brain, and then a few going kind of right at the edge of the cerebrum cerebellum border that I pointed out earlier. Those veins all drain the blood out of the brain, and they bring with it some of the cerebrospinal fluid, which is explained in here. There are 3 layers of connective tissue surrounding the brain. This picture kind of shows them. But these are the 3 names. Okay? The dura mater is a thick, tough connective tissue layer surrounding the entire brain and spinal cord. Okay? The words dura mater translate to tough mother. That's what those words mean. That's kind of what they are. Then the arachnoid, and I have always felt kind of strange calling it the arachnoid mater, because that would translate as spider mother, which is not at all what it is. It's a thin membrane. It's called arachnoid because the deep side of it, the inferior side of it, if you will, has a bunch of fibrous proteins extending away from it, connecting it to the next layer, the pia mater. And that looks kind of like spider webs, and that's where it gets arachnoid from. That space, called the subarachnoid space, is where a lot of cerebrospinal fluid is circulating around the outside of the brain and spinal cord. And then the pia mater, which is a very thin membrane directly covering the outside surface of the brain and spinal cord. That translates to mean thin mother. Okay, so dura mater is tough mother, pia mater is thin mother. It's sort of the 2 layers of connective tissue encasing the brain. The arachnoid's kind of in between it, which provides space for cerebrospinal fluid to circulate around. And this picture down here shows where the cerebrospinal fluid is made, which is in the ventricles. Some is made in the lateral ventricle, some is made in the 3rd ventricle, some is made in the 4th ventricle. The cerebrospinal fluid flows from the ventricles, lateral ventricles in the 3rd through the cerebral aqueduct into the 4th, picking up more and more fluid as it's produced in each of those locations. And then it drains out around the cerebellum into the space of this arachnoid, subarachnoid space that I just mentioned. And it circulates around the outside of the spinal cord in the brain, eventually draining out through these things called granulations that poke through the dura mater and filter the waste products out into the blood to be drained away, which is what this is showing. And it's described in this section here. So I just wanted to point out that circulation stuff, kind of on the heels of looking at the embryological development, because they're mentioned here in this table. The lateral ventricles are in the cerebrum, the 3rd ventricle is in the diencephalon, the cerebral aqueduct passes through the midbrain, the 4th ventricle is in the pons cerebellum area and down into the medulla, and then there's a central canal, which is the hole that goes right down through the middle of the spinal cord. There is some cerebrospinal fluid in the central canal, but it's a dead end. It's a cul-de-sac. There's nowhere it can go from there, really. So that's not very important. And the spinal cord is really thin, so the cerebrospinal fluid on the outside surface in the subarachnoid space does a pretty good job of helping to carry waste products away from that tissue. But there is a central canal there. So I want to talk about the structures of the central nervous system. And for you to understand this, you really need to be looking at these brain models. So I have 6 brain models up front and 5 in the back, I guess, which is close to enough for everybody to be able to look at one, but I think there'll be a few of you that might have to share. So go ahead and grab a brain model and have it in front of you while I talk about this. So first off, remember where you picked yours up from, because at the end of the class when you're putting them back, I want you to put them back where you got them from. The 6 that were up front I got from next door, so I want to be able to put them back next door easily. The 5 that were in the back here actually come out of a cabinet here. I pulled them all out of the cabinet because I didn't want people grabbing a couple of other models that are in here because they're big and clumsy. But for those of you that got your models from the back here, you're going to put them back in this cabinet here when you're done at the end of the day. You'll know which one you got it from because this is room 500 and next door is room 502. And so your brain model will have a 500 or a 502 on it, and that tells you where it goes. So when you're done, the 502s are going to go up front here, so I can take them back next door. 500s are going to go in that open cabinet back there. Now, to begin with, the adult brain is basically separated into 4 major regions, which I've kind of already touched on, but I want you to see where they are now. There's the cerebrum, the diencephalon, the cerebellum, and the brainstem. Okay. Those aren't exactly the words we were using before. The brainstem is actually the midbrain, pons, and medulla all together. Now, there's 2 models that you have, and there are subtle differences between them, which aren't terribly important right now, but I will make some points about the 2 models. There's the hard plastic model and the squishy plastic model. So that's how I distinguish them. I've already explained about the cerebrum. It's the big wrinkly part on the outside, which you can see making up most of the brain. We're going to talk about the cerebrum in more detail and understand where the parts of it are. If you take the model apart, you can see the 2 halves. On the inner surface, the medial surface of the brain, you still see the wrinkly aspect of the cerebrum. It extends here. It ends where there's this sort of C-shaped structure right here. And that's kind of a good boundary of where the cerebrum ends. That C-shaped structure is what we call the corpus callosum. 2 words that both start with C. It represents a very large fiber tract, myelinated axons that go from one side of the brain to the other. And I have a little bit to talk about with that in a second. Inside the curve of that C, what we see down here is the diencephalon. Now, in the squishy brain model, there's a brown circle. In the hard plastic one, there's a little yellow circle. In the hard plastic one, there's also a blue streak at the top. There's not a blue streak on the squishy plastic one. But ignore the blue part on the hard plastic brains. Just the pinkish area that has a little yellow circle, and then the stuff that has a little brown circle on the squishy brain. That's where the diencephalon is. That dot, brown or yellow, whichever one you're looking at, is the little bit of tissue that crosses midline. It made that hole in the 3rd ventricle I showed you a second ago. It is actually where the thalamus is. And the thalamus is about the size of the end of your thumb. So there's a thalamus on the left side and a thalamus on the right side. And they just barely touch right in the middle. And that's where that little circle ends. It's not really a functional connection. It's just the 2 sides of the brain growing kind of push into each other right there. And there's a little bit of a touch there. And that's that dot, which is the hole in the middle of the 3rd ventricle cast. Now, there's sort of a little groove. So if you think about the thumb, and the dot was right about in the middle, slightly forward part of the thumb. If you go anterior, a little bit inferior to the thumb shape, you'll see a little groove, and then a kind of triangular thing pointing out. That triangle has actually kind of got 2 points to it, sort of like the beak of the rooster face I always see when I look at the ventricular caps. That's the hypothalamus. The 2 points are sort of going to this 2 important things. The first point is right above what's called the optic chiasm. That's where the optic nerve turns into the optic tract. And then the bottom point is where it connects to the pituitary gland. Now you might have, and this is true for some hard plastic or squishy plastic brain, you might have a little peanut-shaped thing attached by a wire hanging off of half of your brain. It will not be on both sides, there's only one of them. You might not have one at all, and if you don't worry about it. That's the pituitary gland. Okay. And so if you have it where the little wire part connects back, you can see that's sort of the bottom part of the hypothalamus. Now, this little bit of the model is really fragile, and so it's fallen off more of the models than it's stuck around on. So not everybody will see it, but where it connects is come on, is there a picture? So this is showing the diencephalon, the thalamus, and then the hypothalamus. Oops, sorry. Hypothalamus down here. And it comes to kind of 2 little points. The rear more of the 2 points has the pituitary gland hanging off of it, which not everybody will have a pituitary gland because they fall off these models quite a bit, but there it is. Okay. And the shading that you can see here kind of shows you the extent of the diencephalon. Now the C-shaped structure that I pointed out earlier, this is the corpus callosum here. There's a little bit of a gap between the corpus callosum and the actual edge of the diencephalon there. If you have the hard plastic brain, that's kind of where that blue streaky thing is. If you have the light plastic, the squishy plastic brain, there's not a blue thing there, but you can see a smaller ridge that's right above the diencephalon. The space between the corpus callosum and that ridge is a very thin piece of tissue. If we were looking at actual brains, it might not be visible there. But if it were, you could poke through that very easily. It's just a thin membrane of tissue. And beyond that would be part of the lateral ventricle. Okay, so there's this little curtain of tissue right there on midline separating the 2 lateral ventricles. Very thin. And it fills in the gap between the corpus callosum and the diencephalon there. Okay, so there is a suggestion of that in the model. It's just not quite so clear. But that's the diencephalon. The other parts of the diencephalon, the epithalamus, subthalamus, those kinds of things, we're not going to worry about too much right now. And then we have the brainstem, which is really 3 different pieces: the midbrain, the pons, and the medulla. Now, I was sort of pointing out where the pons was when I was showing you where the cerebellum is. On your model, the easiest way to figure out where the pons is there's a big oval structure on the anterior surface of the ventral surface of the brainstem. If you go back from there to the edge, all of that's the pons, not just the oval and what's next to it is the pons. Now the word pons technically means bridge, and it's named for that oval structure in the front. It is a bridge of white matter that goes from one side of the cerebellum to the other side of the brain. Okay, so sort of the bottom of the pons, the ventral surface that's exposed, is just this big bridge of white matter. That's what you can kind of see there. But really the pons includes the gray matter that's adjacent to that, which we see here. At the top edge of that oval, from that point up to where the diencephalon starts, that's the midbrain. There's a little groove that goes right along the posterior edge of the midbrain. That's the cerebral aqueduct. Okay. I don't quite remember what it looks like in both models, but so if you have the squishy plastic brain, there's the number 34, 32, 31 all around it. There's just a little groove right along there. You look at a hard plastic brain. The hard plastic brain has fewer numbers on it, so it's hard to see. But you can see a little groove. It's just not, there's no numbers pointing to it, but a little groove right there. That groove is passing through the midbrain. Now in this picture, it's a little bit misleading. The way that they've colored this picture, which I didn't catch in time for publication, that groove is right at the edge of the blue color that's shading in the midbrain. But in fact, the midbrain is still some stuff back here. They just didn't quite shade it in correctly. And on the model, that part is a little bit more obvious. There are two bumps right here, okay, sort of where my fingers are. I'm going to mention them by name just because I want to name them for a second, but you are not responsible for knowing the names, but I want to talk about what they are. There's the superior and inferior colliculus. Now the word colliculus in Latin means knee-shaped, so they look like little bumps like knees. And if you're looking at a real brain, you'd get that. But, they're sitting there. The inferior colliculus is part of auditory processing. It's one of the relay stations necessary for information that's coming from your inner ear getting into your brain. That's what it does. The superior colliculus is a place where visual, auditory, and somatosensory information all comes together. It's a really kind of cool part of the brain. If you stick electrodes in and record activity there, you'll find that there are 3 maps of the world in the superior colliculus. One map is visual information, and you can stick electrodes in and record activity when a light shines in this part of this visual field or that part or that part or that part or whatever. It will map out the 2-dimensional visual field that you look at. If you go a little bit deeper into that, the next layer is a map of auditory space. And you can map where your brain perceives sounds coming from. So if you hear a sound coming from back here, or from over here, it'll stimulate different parts of that map. And then the third layer is a map of the surface of your body. So if you're recording there, you can map exactly where something touches the surface of your body. It's important for your body to be able to do what it needs to do to coordinate that information together. A quick example of what that would be like, which is not terribly important for survival, but let's imagine you're walking down the sidewalk here and you walk underneath a tree. Okay. You hear some chirping. Okay. You can hear chirping in the tree up here, and because those 2 maps are lined up, your brain knows to look at that location. It synchronizes auditory and visual space together. Okay. And then if you look up there and you feel something hit you on the head, you know that's poop from the broach track. It's kind of a silly way to illustrate it. You could just as easily tell a story about hearing and feeling a mosquito landing on your skin somewhere. It's still going to use that kind of those maps together. For the human condition, it's not a primary aspect of what's important for us, but just to illustrate it, I like to use a little comparative stuff. This is essentially the frog's main motor. It has a map of its visual space. It has a map of where its tongue goes in space.
if you put any kind of black dot in the visual field of a frog, it'll automatically shoot its tongue out to get that black dot. And it's happening because of the maps in that part of the frog's brain. Frogs don't have cerebrum. They just have this stuff here. So it's very important for how frogs work. It's a fairly minor part of what happens in the human brain, but it still does work quite well, and it's important for coordinating visual, auditory, and somatosensory information together. Just wanted to illustrate that stuff a little bit for you. That's the midbrain. It's about all there is to it, and I'm not going to really get into it at all, but I wanted to tell you something about it. Okay. So that's the brainstem. And so here's a little explanation. Oh, actually in the book here, it does have the inferior colliculus and superior colliculus mentioned. I wrote it, I forgot it was there because truthfully, I don't really ask you any questions about that outside of part 1 or 2 multiple choice type of questions, but the part 3 and 4 questions that you have for this chapter don't deal with the midbrain because it's really a minor part. And then a little something about the pons and the medulla. And then finally, we have the cerebellum, okay, which is the big wrinkly thing on the back of the pons. The cerebellum is largely responsible for directing coordinated movement. Talking about the embryological aspects, I like to stress the idea that anything communicating with the cerebellum has to go along the path that the embryo lays out. And the reason I mention that is because those connections are very important. When you decide to move part of your body, part of your brain up here, which we're going to talk about later on, sends a signal down to your spinal cord to make you move muscles. That signal gets duplicated and sent into this cerebellum. And all of the sensory feedback from your body moving goes up your spinal cord and into your cerebellum. Your cerebellum compares those things, compares the original command and the sensory feedback for how you behave, and makes any adjustments as necessary. The easiest example of that is walking. Okay, so part of your brain up here says, okay, walk, sends the signal down. Walking is actually largely a reflexive activity. And so that one command says, okay, walk, and you start walking. If something throws off your walking, you're not really going to do too much of that consciously. You're not going to correct for that too much consciously. It's happening mostly in the cerebellum. Okay, so 2 examples, one kind of extreme and not one that you tend to run into too often. But let's say your brain says to walk, just basic command to walk, without saying anything about dealing with the fact that you're standing waist deep in water. Okay. So if you just tried to walk normally, but you're waist deep in water, it's going to be really hard to move your legs. So the sensory feedback of the fact that your legs are not moving as much as your brain intended them to move goes back to the cerebellum, and the cerebellum changes the instruction to say, take bigger steps, and so you can move your feet further. Kind of an unusual situation, but it illustrates the idea. Better, more realistic example is, let's say you say your brain says to walk, and you're walking, you're not watching where you go. But then suddenly you step on something, and your foot bends the wrong way. The way that you compensate for that is simply that your cerebellum says, okay, your foot's not where it's supposed to be, so compensate by putting weight on the other foot and get your balance back and keep walking. It's completely reflexive, well, I should say, it's completely subconscious what's happening. You're not consciously saying, oh, I've got to do that. Consciously what happens is about a step or 2 later, you look back and try to figure out what you're doing. Everybody's. But really what's happening in your brain subconsciously is the cerebellum is taking the sensory feedback from how you're walking and the fact that things didn't quite work the way that your brain intended, and it corrects for that and you walk properly. Now, a great way to kind of illustrate what the cerebellum is important for is talking about a clinical test, which is part of the neurological exam in that chapter, which hopefully we'll get to talking about later. But a
I do want to talk about that now. It's called the coordination exam, and it's testing coordinated behavior. I'm sure none of you are familiar with what I'm about to say, but it's more commonly understood as the field sobriety test. So none of you have ever had any experience with the field sobriety test, I'm sure. Field sobriety test, anybody know what it is?
Yes.
Answering that question will not give away the fact that you've been pulled over for DUI. Field sobriety test is what's performed by the state trooper on the side of 91 when you've been driving drunk. Or when
they suspect.
Yeah, when you're allegedly been driving drunk. I'm sorry. But what it is that the cerebellum is very sensitive to alcohol. Okay? And in fact, it's the cells that we looked at, those Purkinje cells that I showed you last week, those are in the cerebellum. Those are very sensitive to alcohol. And those are the main output cells of the cerebellum. When you have alcohol on board, they don't function properly, and the output of the cerebellum's off. So, field sobriety test is the coordination exam, essentially. There's sort of three major components that everybody's probably heard of, thanks to movies and books. There's walking a straight line, and I just explained about walking, so there you go. The cerebellum gets direct input for your sense of balance from your inner ear. And so if you're walking and you're trying to, keep to a straight line, you need to keep some balance. And that's really part of what the cerebellum is doing, getting that equilibrium balance. If you're drunk, it's not going to work. You're going to lose your balance. Another big one is touching your finger to your nose, okay, like this. But there's a couple little twists to that idea. Okay. First off, you have to close your eyes. Okay. Now I'm not using any visual information to get my finger to tip my nose. And then the other thing is you have to tip your head back. Okay. So now my sense of balance is thrown off a little bit, and I'm moving. If you're drunk, it's like this, and you're not going to quite make it. Because the cerebellar output is responsible for driving that non-visual, off-balance version of touching your finger. And then the third one is saying the alphabet backwards. Now, it's not about the cognitive ability to say the alphabet backwards. Not every sober person can do that. Okay. I can get to Q pretty easily because I've been doing it for this lecture plenty of times. But if I try to do all 26 letters, in the end that I haven't really practiced too much in this lecture, I get kind of messed up. Granted, if you're drunk, you'll just be completely off and it'll be pretty obvious. But really what it's testing for is the repetitive, coordinated activity of speech. Now, if this is being done in a clinical setting and you're testing what's called rapid alternating speech, you would be asked to say the 3 syllables la ka pa over and over. La ka pa, la ka pa, la ka pa. Moving the tongue and other muscles around your mouth and your pharynx to make those sounds is alternating between muscle groups that takes coordination. Now, in the field sobriety test, they don't do low, they don't do la ka pa. Kind of ironic that I'm trying to talk and then say all this coordinating movement wrong. But instead, they go with the saying the alphabet backwards because it has built into it those kind of coordinated speech patterns also. Now, it's going backwards because everybody can do the alphabet forwards without using your cerebellum. A, B, C, D, E, F, G. Learning to sing is a coordinated activity that once you're good at it, you no longer need the cerebellum to do. Just like riding a bike. You don't really need the cerebellum once you're riding a bike. You don't need your cerebellum really to walk unless something gets in your back. Saying the alphabet backwards requires you to change things up. So Z, Y, X, W, V, U, T, S, R, Q. Okay, you can go through that too. I got a little messed up. Okay. But being able to do that, to make the change between those consonant sounds is unrehearsed. If you really want to try to beat field sobriety and you try to learn to sing the ABCs backwards, one, they'll probably say, okay, here's somebody who's practiced this, who probably means somebody that needs to beat the field sobriety test, and they're going to have you blow into a tube just the same. You're so drunk that you're not going to be able to do it anyways. So, but that's what the cerebellum's about. So I just went through the 4 major parts. Okay, I mentioned the cerebellum, but we're going to talk about that in more depth in a little bit, and we'll come back to that. The diencephalon, thalamus and hypothalamus primarily, the brainstem, midbrain, pons, and medulla, and then the cerebellum. Okay, the rest of the central nervous system, the spinal cord, and we looked at a cross-section of the spinal cord last week. Here's a picture kind of similar to what we saw. I said this briefly earlier, but if we sort of split the spinal cord right here at the middle part, midline is a vertical line here, but if we separate posterior from anterior, we're basically separating the sensory portions from the motor portions. And the cells we looked at last week were in the ventral or anterior gray matter, ventral horn, and they're responsible for actually moving muscles, okay, which we see here. We call the gray matter in the spinal cord horns. Okay. So there's the ventral or anterior horn and the dorsal or posterior horn. Ventral horn controls muscles. The dorsal horn processes sensory information. Okay. And then we also have white columns. So the white matter that's on sort of the outside of everything is segregated into a few different regions. Now, most obviously between the dorsal horns, there's a completely isolated region of white matter, which is just called the dorsal horns. And then on either side, we would have the lateral, sorry, I said it wrong. Between the dorsal horns, there's an isolated region of white matter called the dorsal columns. So the misfocus. And then on the sides, we have the lateral columns. And then on the anterior side or ventral side, we have the anterior columns. Now they're called columns because if you look at the entire spinal cord, it does look like columns of white matter going the length of the spinal cord. And so this is an anterior view here. So we're looking at the anterior columns of the spinal cord up there. And then on this torso model here, we have now the posterior section view here. So we're looking at the posterior or dorsal columns running up here. And then between the anterior columns that are visible in that model across the way and the dorsal columns visible in this model here, on either side we have the lateral columns. The white matter is separated up into specific bundles of fibers that are going different places, which there's not a picture of right here. I want to show you some of those, which I'm going to talk about in the second half today. After I talk about some other things, bless you. So we'll come back to the idea of where the columns are, but the basic layout. Most of the outer surface of the spinal cord is white matter, and especially looking at the whole spinal cord, we can see these columns running there. And then the gray matter inside makes this kind of H or butterfly, or talked about the kissing bunny heads last week. Whatever ink blot interpretation you want to make of what it looks like inside. They're primarily separated into the dorsal and ventral columns. And it's really just kind of convention to use dorsal and ventral rather than posterior and anterior. So we stick with those. Now in this picture, more so in the drawing than in the drawing up above it, you can see a little pointy thing coming off to the side here and another pointy thing there. That's a third horn. It's what's called the lateral horn. And I want to point it out here, since we're talking about the structure of the spinal cord, so that you know that it exists. But what that's responsible for, we're going to talk about next semester. So file away in your mind that there is a lateral horn, and what it basically is responsible for is controlling non-skeletal muscle responses. Okay, so the ventral horn controls skeletal muscles. The lateral horn controls cardiac and smooth muscle and glandular tissue, which is the autonomic system. Okay, so when we talk about the autonomic system in AMP2, the lateral horn gray matter is going to be important for that. That's an overview of all of the brain, adult nervous system, brain and spinal cord. What I want to do with the rest of the time that we have today is to talk more specifically about the cerebrum. We're going to start by talking about the surface structures, what we can see on the surface of the brain. And then I have a couple of things I want to talk about what goes on deep inside of the brain. But before I get to talking about the surface of the brain, I want to talk about this structure called the corpus callosum. So in this picture here, let me just enlarge it a little bit. The dotted line that we see superimposed on lateral view is supposed to show you where that C-shaped structure that I pointed out before is. And then the label corpus callosum you can see up here. This is white matter that connects the right and left sides of the cerebrum. Now, you've probably heard of the right and left hemispheres of the brain. You might have heard reference to people being right-brained or left-brained or some bullshit like that, excuse my language. The reason I call it bullshit, and I don't really excuse my language when I use it in that context, is it's not true. Okay? If you've ever seen or heard anything talking about people being right-brained or left-brained, it's not true. People are whole-brained, except for people that are half-brained. And by that, I mean people that are the basis of the data that made everybody think that there's such a thing as right and left brain. There's a condition called epilepsy, which is when neurons start to fire independently by accident. Let's say a neuron right here fires off an action potential, and it connects to a neuron here that fires off an action potential, and it connects to one here that fires off. So suddenly you get activity building up here, and then that activity spreads out further and further, and the whole brain ends up being incorporated into this epileptic activity. It's a disease state, and it can result in serious seizures. And if the activity is going to incorporate motor control centers of your brain, you're going to have grand mal seizures. You're going to lose control of your body. All sorts of horrible things can happen. When they're really severe and they start on one side of the brain and they spread to the other side of the brain, and it happens regularly, once a day or more, it's impossible to live like that. It's also impossible to cure. So in that situation where the quality of life is so degraded by the condition that the person really can't do anything. One way to solve that is to do a hemispherectomy. Well, actually, that's not the best word for it. Corpus callosumectomy? I don't know. Actually, I shouldn't say ectomy anymore. The ecto in ectomy means out, so to cut something out. So I guess it would be more appropriately called the corpus callosotomy. It sounds too much like colostomy. But anyways, the point I'm making here is you can cut through the corpus callosum. That's the only way that the right hemisphere and left hemispheres of the cerebrum communicate back and forth. And if somebody has intractable epilepsy, which means, severe seizures that affect the entire brain, they go into grand mal seizures, and it happens very often. The intractable means It's so far gone, there's nothing to do about it. You can cut through the corpus callosum and you'll see an immediate change in the quality of life for the patient. Now, I want to end the first half today with a story about that. So I'm going into the video content part of the Blackboard website, and so I have a video down here which I want to play for you. Now, you might not be able to see just yet. I'm going to turn off the light in a second, but there are two people sitting there. The one that's in the foreground is Alan Alda. What we're looking at here is a video. It's actually one part of an episode of a PBS special. I can't remember exactly what it was called now, but Alan Alda hosted this series of shows. I don't remember if they're just about the nervous system or if they're about various physiology aspects of the whole body. But this one episode deals with the split brain issue, cutting corpus callosum. So let me play this for you. And I have the mute on. Oh. Nope, that didn't do the trick. Okay, I'm not going to go through all that. Well, actually, I am.
We began our journey into the human brain here on the campus of Dartmouth College in New Hampshire. I'd come to meet one of the world's leading brain scientists, Mike Gazzaniga, and a man he's worked with for over a decade. A man with two brains. You've been working a long time with Dr. Gazzaniga?
14 or 15 years? It doesn't seem like that long, does it? Oh, no.
The collaboration began when Joe had surgery.
And you had this procedure to correct an epileptic problem, is that right? Trying to stop the seizures. I was having seizures like every day, so there's 2 or 3 a day.
To control Joe's epileptic seizures, the surgeon severed the connection between the two halves of his brain. Cutting the corpus callosum like this prevented the spread of the electric storms that caused his seizures. But it also prevented the left and right halves of his brain from communicating with each other. In the years since the operation, Joe's epilepsy has been under control. He now earns a living at an egg farm, and in his everyday life, he's largely unaffected by the fact that his left and his right brains work independently.
Do you feel any different when you think about something than you did any differently from what you felt before the procedure? Better back up, right? That's something everybody could use.
I found out how true that was right away when I was asked to draw a different shape with each hand. In a brain like mine, roughly speaking normal, at least all in one piece, the left half of my brain controls the right side of my body, while the right brain controls the left side.
Oh, no.
But because the two halves are connected,
Nothing wrong with that.
getting each hand to work independently isn't easy.
Well, we're saying that the fact that Alan's hemispheres are connected, and that the motor messages from one are confusing the motor messages of the other.
I was just drawing an upside down duck. Okay? But when Joe is given the same task, his two hands operate as if controlled by two separate brains. What's happening is that each half of Joe's brain is given a separate instruction. He's asked to fix his eyes on the cross in the center of the screen. Anything flashed to the right of the cross goes only to his left hemisphere. Things to the left go to his right hemisphere. Because the two don't communicate, each hand does only what its half of the brain sees. It's really like two different people doing the same.
That's right. Okay, that's the idea. Okay, Joe, I want you to keep your hands up.
In an experiment that's now a classic in brain research, Mike Gazzaniga over 30 years ago used a similar setup to find out if the two halves of the brain are specialized to do different things.
Sure.
Joe is being flashed a word only to one half of his brain. Words flashed to the right are seen only by his left brain. And Joe can report seeing those words just fine.
Good.
But when a word is flashed to his right brain,
Didn't see that. Okay. John, we ask,
but now watch what happens.
draw that with your left hand.
I'm gonna be lost.
Why don't you try drawing another picture of it over here? That'll help you.
Oh,
Oh, okay.
it's almost as though somebody has given him a secret communication.
That's right.
Now he knows that is up until when he was blind.
Exactly.
When Gazzaniga first did this experiment, it instantly proved that the ability to speak resides almost exclusively in the left hemisphere. Not until he sees what his right brain is drawing is Joe able to name it.
He said church after looking at it. But he had to figure it out about as long as we did. It's really interesting.
It's a picture here of somebody communicating almost with another person.
Communication is not occurring inside the head, it's occurring out on a piece of paper.
Yeah.
Wow. You want to draw a little bit more?
So far, Joe has been seeing only one word. So things get even stranger when he's flashed two words, each to only one half of his brain.
The right hemisphere is on toad.
Yeah. And so his left hand draws a toad.
So there's the toad.
Oh, it's a toad. And this time I was able to guess what was coming.
Now put a little three-legged stool in there.
Joe's speaking left brain saw a stool. Saying the word lets the hand that's controlled by his right brain in on the secret. That's great. That's really interesting. And if he had seen that with the corpus callosum intact, he would have drawn a toadstool.
Right.
Not a toad and a stool.
Right, exactly. I've been doing this for 35 years. Yeah.
And it
gets me every time.
Yeah.
So that's a quick little show of some of that stuff. Let's go ahead and take a break, and then we'll come back and I'll talk a little bit about some of the things that came up in that video, and we'll go from there to talk about what we can see of the cerebrum and what that means about function. So by that clock, about quarter after, come back in here.
Do you want these back?
No, we're going to need them in a second.